Okay, so in lecture 10, we built our heme, all right? Remember, heme is composed of your protoporphyrin ring 9 and iron. All right, so now what do we have left to make the hemoglobin molecule? We have heme, we need globin, right? Okay, so now we're going to build our globin chains. I have a video. I might show that. I'll show that at the end, I think. All right, so now we're going to build our globin chains. And keep in mind this molecule right here, all right? So remember, your globin chains are going to be composed of an alpha chain and then some type of non-alpha chain, all right? That's your pair. Each hemoglobin molecule will have two pairs, okay? So in this example, we have our alpha chain and beta chain, all right? Combined with another alpha chain and beta chain, okay? So you have two pairs of globin chains, an alpha chain and a non-alpha chain. Okay? We're going to talk about the different types of chains. All right, so globin chain synthesis occurs in the red cell ribosomes during your pronormoblast all the way to your reticulocyte. All right, so remember the cell has a nucleus, protein synthesis is occurring. And so that is when all of these globin chains are being produced. All right, so the globin synthesis is actually going to be directed by 8 different genetic loci per haploid genome. And these are going to be, this is going to result in 7 different types of globin chains. All right, so you have your zeta, your epsilon, your gamma A, your gamma G, your delta, beta, and alpha. Okay, those are all the different types of chains. So remember I said that your globin chain is composed of an alpha and a non-alpha, right? So your alpha chains will be in every type of globin, all right? It's always gonna have an alpha chain. So your alpha-like is also zeta, all right? So alpha-like would be, could be alpha or zeta. We're gonna talk about zeta is present in the embryonic formation. About 10 to 12 weeks gestation, zeta is not going to be produced anymore. All right, so it's only during fetal development that we'll ever see zeta. And actually epsilon too is during fetal. All right, so this does use Greek letters. So if you guys aren't familiar with the Greek letters, it does prove to be challenging. So make sure you study up on that as well. And so here's your globin chains: zeta, epsilon, gamma, delta, beta, and alpha. So these chains are going to combine to form different types of hemoglobins. All right, so our most predominant adult type of hemoglobin is hemoglobin A, which is comprised of 2 alpha chains, 2 beta chains. All right, so that's how it works. That's our normal adult hemoglobin A. And then in fetal hemoglobin, you'll have 2 alpha chains and 2 gamma chains. That composes adult or hemoglobin F. All right, and we're going to go into detail as well, but that kind of lets you know how these chains combine. All right, so just to let you know where this is happening on the chromosome level, it's occurring either on chromosome 11 or 16. So your alpha and zeta is present on chromosome 16 on the short arm, so on your P arm of chromosome, and then all of your other chains come from chromosome 11 on the P arm of chromosome 11. All right, so remember chromosome 16, your zeta and alpha, and then chromosome 11 will control epsilon, gamma, delta, and beta. So it's actually DNA synthesis that controls the production of these globin chains. Transcription, translation, codons are involved in DNA replication. So while one globin chain is being produced, the other chains are being suppressed. So for example, if beta chains are being produced, only beta chains are being produced, all the other chains are being suppressed, right? So it has to all do with DNA transcription and things like that, all right? So it's all protein synthesis. right, so remember I mentioned that zeta is your embryonic alpha-like chain. So zeta is going to start out, and let me just go to my chart right here. All right, so here's your zeta, right? Notice that it's going to start out during fetal development, and this is weeks of gestation right here. Right, so your zeta and epsilon are going to start out till about 10 to 12 weeks. They're going to no longer be produced, and then your alpha and your gamma chains are going to start being produced. All right, so notice that you always have alpha. All right, your alpha is always going to be there, and it combined with your zeta, or sorry, your epsilon, and it will form Gower1, which is a intrauterine. Do I have that chart? Oh, sorry, let me back up. All right, so zeta and epsilon are your embryonic globin chains. They will form during the first like 10 weeks of gestation, they will form Gower1, all right? And then once zeta is no longer produced, alpha is going to take over. Once alpha takes over, it will be produced from here on out for the rest of the individual's life, okay? And then you'll have gamma. So this is why once alpha and gamma are formed, 2 alpha chains, 2 gamma chains, that forms our hemoglobin F. Hemoglobin F is our fetal hemoglobin, okay? And then beta chains will also start being produced. And then once alpha and beta chains combine, then we have our normal adult hemoglobin of hemoglobin A, okay? And then you also have delta that's going to start being produced right before birth. And alpha can combine with delta to form another one of our normal adult hemoglobins. It's hemoglobin A2, okay? So these are the most common types of hemoglobins that we'll study, okay? Just remember, you'll have 2 alpha chains and then 2 non-alpha chains, whether that be gamma, delta, or beta. Okay? So remember, 2 alpha chains, 2 gamma chains form our hemoglobin F. That will start producing about the 3rd month of fetal development, and then up until 1 year after birth, it is normal to have hemoglobin F. After that point, hemoglobin F should drop off to like less than 2%, I think, for a normal adult to have hemoglobin F levels. And then it should be like 95% hemoglobin A for adults, okay? Right, and just like looked at that chart, beta chain production is going to rise gradually, and it's going to reach adult percentages between 3 to 6 months is when the alpha chains and beta chains should be at their peak. And again, so you'll have 2 alpha chains and 2 non-alpha chains. All right, so here's your normal adult hemoglobins. It's very important that you know which chains compose which hemoglobin and then what's their presence in adults versus newborns, okay? So our hemoglobin A is comprised of 2 alpha chains and 2 beta chains, and it should be about greater than 95% in an adult. You'll have about 10 to 40% in newborns up to 2 years. Okay, so it's kind of an inverse situation, okay? So when newborns are first born, their predominant hemoglobin is going to be hemoglobin F, which is fetal hemoglobin, comprised of 2 alpha chains, 2 gamma chains. And they should have anywhere from 60 to 90% hemoglobin F right after birth. And then you have your hemoglobin A2, which is 2 alpha chains and 2 delta chains. Does that make sense to everybody how these chains are combining together to form our hemoglobins? Okay. And again, very important that you guys know the amounts. So at birth, hemoglobin F is going to be the predominant hemoglobin present, should constitute about 60 to 90% of the hemoglobin. As the individual ages, all right, so as the newborn ages, hemoglobin F is going to decline, and then it'll have about 7% at 12 weeks after birth. All right, so it's slowly decreasing, and then by 2 years at the max, that hemoglobin F should be less than 2%. Okay. So there are some conditions that we'll talk about, such as thalassemia, when you have a decreased production of either alpha chains or beta chains, or an absence of alpha or beta chains. So alpha is going to bind to another chain, a non-alpha chain. So in the event beta is not present, so if you have beta thalassemia, then that means you have a decreased amount of beta chains being produced or an absence of beta chains being produced. Alpha is going to bind to either your gamma or your delta. All right, so then in those instances, you'll have increased amounts of other hemoglobins. And the way we can do that is a hemoglobin electrophoresis to detect that. All right, so that's what's coming. That's how we're going to identify some of these different types of thalassemias and hemoglobinopathies. Okay.
What type of symptoms would they have without the beta chains?
So it depends. If you take a look at what's a common factor in all of these hemoglobins. What type of chains? Alpha. What happens if you don't have any alpha chains? If you have a 4-gene deletion, meaning that there's no alpha chains being produced, it's not sustainable for life. All right, it's called a 4-gene deletion, no alpha chains, hydrops fetalis. Some of you guys might have heard of. The fetus might be born, but he will not last very long. All right, so that's the most severe case, and that would be severe, what we would see on the peripheral smear, severe hypochromatic microcytic cells. They would probably know intrauterine because that baby's going to be in distress. Yeah.
So what happens if they notice that the baby doesn't have alpha chains?
So you have some, remember we talked about zeta and epsilon chains? You have those being produced while the baby is in uterine. And so they will be produced. They're not as well as, they don't do a good job as carrying oxygen as our normal alpha combined with a non-alpha chain. So the baby will be okay intrauterine. But then once those zeta and epsilon chains aren't produced anymore, they won't be able to carry oxygen. So they'll have cyanosis, they won't be able to breathe, and that's what causes them to expire.
Will they know before the baby's born?
They should, because once those epsilon and zeta chains drop off, the baby's gonna not have adequate oxygen supply. Now, mom's oxygen supply can help a little bit, but not first. I don't know if they would know intrauterine, probably because that baby's going to be in a lot of distress. Probably be anemic.
They usually born like premature.
I think so, yeah. And then mom too, she'll probably have like premature cramping and yeah.
They had to do weekly ultrasounds.
So there's also another type of thalassemia, beta. Now, when we talk about the 4-gene deletion, that is the most serious case. You could have a single gene deletion where you're still producing some alpha chains. That's not as serious. You'll still live, you just might be a natural anemic. Like that'll be your normal for you. You'll have hyperchromic microcytic cells on a daily basis, but that's your normal. You'll still lead a functioning life. Sometimes, and then you can have a 2-gene or 3-gene deletion, and we'll get into all of that a little bit later. And so it gets more serious. Some, if you have like a 2 or 3-gene deletion, you might have to have multiple transfusions. So to replace the abnormal hemoglobin with regular hemoglobin. So. right, so normal hemoglobin F levels, I took this from your textbook. You guys might have some study questions about this, but so at birth, that hemoglobin F is going to be about 60 to 90%. After 12 weeks, it's going to start declining, all right? It's going to decrease to about 7%. 4 to 5 months, it should be between 1 to 5%. And then by adulthood, it should be that 1 to 2% that we should see, all right? That's in a normal situation. So if you're evaluating the amount of hemoglobin F based on a patient's age, this chart will help you, okay? What if you had an adult with 75% hemoglobin F? You might suspect some type of beta chain abnormality, okay? We have a condition, it's called hereditary persistence of fetal hemoglobin, HPFH, where the patient doesn't produce any other type of hemoglobin. So it's an adult, he has about 75% hemoglobin F. It's just a known condition. Hemoglobin F has a high affinity for oxygen, so oftentimes his oxygen delivery to his tissues is not as good as hemoglobin A or A2. So oftentimes that's an issue for him. But he can lead a normal life. The only thing is if we do hemoglobin electrophoresis on him, he'll have an abnormal pattern. All right, so you have these globin chains, you have your alpha chains and a non-alpha. They will bind together. Here's our famous schematic of our globin chains. They will twist in a helical shape and they're gonna coil like a pretzel, right? And the object of that is to protect their heme molecule, to protect that iron, right? That's their ultimate goal. And again, remember, it's the hemoglobin that comprises the majority of that cell. That's what's going to give the cell its color and its weight. So if you think about the composition of these globin chains, it's all amino acid. Remember, it's protein synthesis, amino acids. So the amino acid sequence is very important. If one single rearrangement in the amino acid occurs, you will have a different type of globin and hemoglobin being produced, all right? This is the basis behind our hemoglobinopathies, such as sickle cell, where you have the formation of hemoglobin S, hemoglobin C, hemoglobin E. Those are all different single rearrangements of an amino acid in the globin chain synthesis, okay? And so when those globin chains, amino acid sequence changes, it changes the shape of those globin chains, and that is hence why the globin chains such as hemoglobin S for sickle cell, they kind of fold on themselves, causing the cells to form a tactoid polymer, hence why we see sickle cells on the peripheral smear, and they are not very well at carrying oxygen, okay? And so tactoid form of cells, that's why if the patient ever goes into a decreased oxygen tension level, then those cells are going to sickle. That's what causes the formation of those cells, all right? It's due to one amino acid rearrangement, okay? Yes.
Does that rearrangement happen to one type of hemoglobin more than the other? Like, does that say this rearrangement is something that happened to our hemoglobin, what, alpha or betas, or you know what I'm saying?
Yeah, so it's on the amino acid level. So you'll have, I think, so like for sickle cell, you'll have a valine substitution in the glycine when it should have been glycine. And so it's just one single amino acid, and that won't affect every cell because, you know, every cell won't sickle, but in the majority of those patient cells, that amino acid rearrangement occurred.
Are we going to find hemoglobin S as, sorry.
So you're saying which one's more common?
I guess.
The one we'll see most common is hemoglobin S. That's our most common one. Hemoglobin E and hemoglobin C. And then it also has to do with your ethnicity as well. So like hemoglobin C is found more in Mediterranean backgrounds. Hemoglobin E is found more in Asians, I think. So it has to do with your hereditary as well. And we'll get more into that as we start talking about our hemoglobinopathies. Okay, so the primary function, so we have our globin chains now, we have our heme molecule, we have our hemoglobin molecule. So remember, the primary function of hemoglobin is to deliver and release oxygen to the tissues. And then it's also going to facilitate carbon dioxide excretion as well. One of the most important controls of the hemoglobin affinity is the 2,3-diphosphoglycerate. You guys remember that? What shunt is it produced in? I heard a lot of things. So 2,3-DPG or BPG used interchangeable. They are the same thing. Think about your Embden-Meyerhof glycolytic pathway. There were some off shunts. Does anybody remember what 2,3-BPG is produced in? Which one? Yeah, the Rappaport luberine. Okay. So you guys need to know that by Friday. All right. This exam, I would not say this exam is hard. It just covers a lot of foundational material needed for red cell metabolism. Okay, so think about 2,3-BPG, 2,3-DPG. It kind of acts as a bridge, okay? It's going to be between those beta chains, and it forms like a salt ionic bridge, okay? And it's going to widen the space between those beta chains. When 2,3-BPG is present, it cannot transport oxygen, all right? So what's going to happen as one of the oxygen molecules is incorporated into the globin or the heme, it's going to break that 2,3-BPG bridge. So it's 2,3-BPG is not big enough to expand once oxygen is incorporated. So, and if you guys know how the hemoglobin molecule works, once one molecule of oxygen is added, the 3 others are going to join it rapidly, okay? So once it's oxygenated, it wants to be at full capacity, okay? And so then the 2,3-BPG bridges is going to be broken, okay? When 2,3-BPG is incorporated into the cell, we call that a tense state. Okay, so it's not oxygenated, it's called deoxygenated. Once oxygen is bound, what's going to happen is that 2,3-BPG salt ion bridge is going to be broken. It's going to have a 15-degree change in its structure. It's going to kind of rotate itself. All right, then that is called the relaxed phase. All right, it is fully oxygenated, ready to travel to the tissue and carry that oxygen to the tissues. Okay. Once it's fully oxygenated, that is termed oxyhemoglobin. An example of that, this is occurring inside the lungs, right where these cells are obtaining oxygen, okay? All right, so deoxyhemoglobin, where 2,3-DPG is incorporated, has a low affinity for oxygen. It readily releases it, okay? Oxyhemoglobin has a higher affinity for oxygen. Those salt bridges are broken, beta chains are pulled together. That 2,3-BPG has been broken, okay? It's no longer there. I know you're like, what all does that mean? Okay, we're getting there, I promise. There's a point. Hemoglobin affinity for oxygen determines the proportion of oxygen that is released to the tissues or loaded onto the cell at a given pressure. Okay, so this is your partial O2 or your PO2 level. How many of you guys have heard of the hemoglobin dissociation curve? Have you guys ever heard of that before? That's what we're working our way to, okay? Increased oxygen affinity means that hemoglobin has a high affinity for oxygen. It means that it does not, it's loving, right? It's loving that oxygen. It does not readily want to release it. And then decreased oxygen affinity means that the hemoglobin has a low affinity for oxygen. It wants to get rid of that oxygen, such as would occur in our tissues. Okay. So the oxygen affinity of hemoglobin is usually expressed as a partial O2 level at which 50% of the hemoglobin is saturated with oxygen. So in a normal person, the 50% saturation usually occurs at about 26, 27 millimeters of mercury, okay? So if you plot your hemoglobin oxygen saturation versus your partial pressure of oxygen, you get a sigmoid curve known as your hemoglobin oxygen dissociation curve, okay? Right here. So this is your hemoglobin oxygen dissociation curve. It's found in your textbook. And it's a physiological curve, sigmoid shape. So if you think about this curve, don't look at it like you do a normal X and Y axis, all right? Saturation is occurring in the lungs, all right? So 100% saturation is going to occur at the top of the chart. Remember, you want 50% of that oxygen to be released in a normal situation at about 27 millimeters of mercury. All right, so noted in your A right here, noted right here by that red line. So your P50 value, your partial oxygen at 50% saturation, normal situations is about 26 to 27 millimeters of mercury, okay? Now what happens if you have a right shift in that curve, meaning that these cells readily release oxygen quicker, all right? So you're going to have a increased amount of mercury at your 50% saturation, right? So notice C by your right shift, indicated by the line C here, is occurring higher than your normal hemoglobin oxygen dissociation curve, all right? So some things that could cause a right shift, if you have increased body temperature, right? So let's say your patient has a fever, acidosis, if there is a pH change in your blood, and the presence of 2,3-BPG. So remember, 2,3-BPG presence readily allows for oxygen to be released, okay? So those are some things that will cause a right shift in that curve. Versus a left shift, notice that your oxygen dissociation curve, you have a lower amount of mercury, millimeters per mercury. Okay, so the opposite of that. So if you have a alkaline pH present in your blood and a decreased amount of 2,3-BPG will cause a left shift in that curve. Okay. Does that make sense to everybody? Some of you guys indicated that you had seen this before. Yes.
We're using this millimeters of mercury, is that blood pressure?
It's not blood pressure, but it's like pulse ox, if that makes sense. So you're measuring like an arterial, you can do an arterial or venous stick and get your oxygen saturation level. Thinking about your question and see if I can word it differently. Hold on just a minute. So you're measuring the pressure of oxygen at a given point in time.
The pressure that's coming off of hemoglobin, like the outward pressure of the oxygen.
Mm-hmm, yeah.
That we're saying here.
Yeah. I don't think it's necessarily oxygen tension, but it's measuring pressure. If that makes sense.
The more oxygen is on the hemoglobin molecule, tend to want to give it up, just you have a higher pressure.
Right.
Assuming that it's not bound up to
Right, yeah,
That what an arterial stick does?
yes. So if any of you guys are ever involved in arterial sticks, this is what they will measure. Yeah, so they usually take them right here. But it can be done venous too. They just have different reference ranges. All right, any other questions on that? All right, so just remember, normally in the lungs, the oxygen tension is 100% saturated, right? The hemoglobin molecule is going to be fully oxygenated, 4 molecules of oxygen. And again, if you look at your chart, this is in your lungs right here, so you have about 100 saturation. And then as the red blood cells circulate, oxygen is released when your partial O2 drops to 40 millimeters of mercury. And that should be, in a normal situation, should be about 25% oxygen that has been released. And again, so normally your P50 value is at about 26 to 27 millimeters of mercury. All right, so we talked about a right shift. So remember, if that curve is shifted to the right, so our tissue is, our cells are releasing oxygen at a quicker pace, all right? What causes that right shift? Hypoxia. All right, so if your tissues are in dire need of oxygen, then the cells are going to be more readily to release it. Increased 2,3-BPG. All right, so remember, increased 2,3-BPG will also, what's the right word? Increase the amount of oxygen release. Some types of anemia, right? Because you have a decreased amount of red cells in anemia, therefore oxygen is not being delivered adequately, right? So your tissues are going to need more. So then the cells are going to be more readily likely to release that oxygen. And then acidosis and increased temperature if your patient has a fever, right? That's your right shift. Opposite of that, your left shift, right? So that just means that your cells are holding on to the oxygen longer, all right? Decreased 2,3-BPG, an alkaline pH, increased quantity of abnormal hemoglobins, and multiple transfusions. And the reason for multiple transfusions is, and we'll talk about this next semester in blood bank, think about donor units. After they're collected, their anticoagulant is added, preservatives are added. The longer those cells sit on the shelf, those cells lose their 2,3-BPG. So they are not readily available. They're not readily available to allow for that release of oxygen. All right. So that's why if a patient has had multiple transfusions, they'll have decreased 2,3-BPG. Okay. And then fetal hemoglobin has a very high affinity for oxygen. It is not a very adequate oxygen deliverer. That's your hemoglobin F, fetal hemoglobin. And then just to break that down, left shift, your pH is going to increase, your temperature decreases, the presence of 2,3-BPG is decreased, versus your right shift, pH is going to decrease, temperatures increase, so your patient could be feverish, and 2,3-BPG increases, okay? That is our hemoglobin association, hemoglobin oxygen dissociation curve. We have some acquired non-functional hemoglobins, and this is just where the hemoglobin molecule has been altered. This does affect the ability of these molecules to carry oxygen. So this will result in hypoxia, meaning that there's inadequate supply of oxygen to the tissues. These patients will experience cyanosis, meaning that they have their extremities will turn blue, they'll start tingling, their lips might turn blue as well. And that's just because they have a high presence of deoxyhemoglobin. And deoxy just means that the hemoglobin molecule cannot bind to oxygen. All right, so there's actually 3 of these that you guys need to be aware of: carboxyhemoglobin, methemoglobin, and sulfhemoglobin. Carboxyhemoglobin occurs when the hemoglobin is exposed to carbon monoxide. So if any of you guys ever heard of carbon monoxide poisoning, this is what happens. The carbon monoxide is going to interfere in the hemoglobin structure, and it's going to prohibit the molecule from being able to bind to oxygen. So if carboxyhemoglobin is present, it is actually 200 times greater for carbon monoxide than oxygen. So it's going to be carrying carbon monoxide instead of oxygen. All right, and this is a fatal condition. Oftentimes you'll see this in smokers. Smokers can have up to a 12% level of carboxyhemoglobin, and that's just because the sulf is incorporated into their hemoglobin. Smokers are used to it. How they compensate for that is they're going to have what we call relative polycythemia. So they're going to have an increased hematocrit, increased red cell mass. It's going to make them appear, they're going to have more red cells to improve their oxygen carrying capabilities. That's what happens in smokers. So then we have methemoglobin, the hemoglobin with the iron in the ferric state. All right, so remember, our iron has to be in ferrous state in order to bind oxygen. So when the iron is in the ferric state, it's incapable of binding oxygen. And so you guys remember, we're going to go back to our glycolytic pathway or enzymatic pathway and the methemoglobin reductase pathway. It is responsible for converting ferric iron to ferrous iron to prevent the formation of methemoglobin. All right, so if there's any defect in that pathway, we will have methemoglobin as well. When this would occur, certain pesticides, antibiotics, they can cause this as well, the formation of methemoglobin. Infants are more susceptible to the methemoglobin than adults just because their hemoglobin F more readily converts, easier converts. Oftentimes, if a patient has methemoglobin, dyshemoglobin, dysfunctional hemoglobin is what it's called, they have like a brownish tinge to their plasma as well, so you'll see that. And then you have sulfhemoglobin. This is an irreversible change. While the other two, you can probably give your patient 100% oxygen and help, this one is irreversible, okay? And what happens is a sulfur atom combines at each of the 4 heme groups. It is going to bind to the oxygen, therefore inhibiting the release of oxygen. I believe sulfhemoglobin, if a patient has this, then their plasma is going to be cherry red. This is not normal. So this would be, you'd have to have some type of exposure. I think anti-malaria drugs are known to do this. It all has to do in how your body metabolizes these drugs. Pesticides are known to cause this. So this is not normal at all. It could, yeah. It probably would be a lot more prominent than hemolysis, depending on the degree of hemolysis going on. But yeah. And due to the addition of that sulfur, the hemoglobin molecule is only going to have a 1 to 100 affinity for oxygen. So that's really decreased binding capacity for the oxygen.
Your body automatically provide or get more red blood cells then?
I think your body would try to compensate for it, depending on the exposure, like what type of drug, pesticide, how serious it was. Then it's probably going to be incorporated into the new cells as well. And then again, your patient's going to have cyanosis. They'll have very extreme bluish coloration in their extremities. Their skin might even have a bluish tinge to it. I mean, that's just due to the lack of oxygen.
So what would happen with patients with sulfhemoglobin? They have to get multiple.
They will ultimately probably, if 100% oxygen doesn't help them, then they will probably ultimately expire due to lack of oxygen. Because even if you give them 100% oxygen, that salt molecule that's incorporated into the hemoglobin is not going to allow for oxygen to be incorporated, right? The oxygen might be there, but it's unable to bind. And so that's the problem. Okay, so erythrocyte destruction, senescence, all of you guys know this already. During the 120-day lifespan, the red cell undergoes senescence, all right? So at the end of its lifespan, it's going to eventually be destroyed either by the spleen or possibly intracellular. The aging is going to be characterized by a decrease in our energy, so the amount of ATP. Remember, the cell has no nucleus, so it has to live with whatever has already been produced before that nucleus was extruded from the cell, okay? It's going to lose its ability to maintain shape, not going to be able to deform, and also not going to be able to maintain that membrane integrity. So what's going to happen? You're going to have a decreased amount of your ATP. Cells are going to be removed by the spleen. What happens to our hemoglobin when the cells are removed from the spleen? The body's going to try to reuse as much as it can. All right, so transferrin is our carrier protein, and it's going to bind iron, and it's going to transport it back to the bone marrow so it can be incorporated into new cells. The globin is going to be broken down into amino acids. They're going to be transported back to the amino acid pool to be reused. That protoporphyrin ring 9 is actually going to be broken down into toxic bilirubin, all right? So albumin is our carrier protein. It will carry that bilirubin to the liver so it can be conjugated and excreted through stool and urine. And that conjugation of the bilirubin is going to occur in the liver. When we start talking about cell hemolysis, intravascular and extravascular, we will go in depth into the process of the breakdown of that protoporphyrin ring 9 and how it's converted to urobilinogen and conjugated bilirubin. Conjugated, once it's conjugated, once bilirubin becomes conjugated in the liver, it can be traveled on down to the intestines, all right, where the gut bacteria will break it down into urobilinogen, which is excretable. So it's normal to see urobilinogen in the stool. You can also pick it up in the urine. So if there's any increased amount of intravascular hemolysis, you might see an excess amount of urobilinogen. That's a test that you guys will talk about in body fluids as well. So about 90% of your erythrocyte destruction is going to occur extravascular, meaning that it's going to occur inside your spleen. And the liver and the bone marrow can be involved too, but most of it's going to occur in the spleen. 10% can occur inside the vessel, intravascular. And this is just when the red cell ruptures in the blood vessels, hemoglobin is going to be released directly into the bloodstream. We have to have methods in place. So if our red cells are destroyed intravascular, it's our carrier proteins that will, transferrin is going to bind to the iron, haptoglobin is going to bind to the globin chains, and then albumin is going to bind to that bilirubin to make sure that they are excreted appropriately, okay? And again, we have a lecture coming up that talks extensively about that as well. So in the event of intravascular hemolysis, after the red cells are ruptured, the alpha and beta chains, all right, those are dimers or amino acids, they're going to be picked up by haptoglobin. Haptoglobin is going to transport them back to the amino acid, okay? But we don't want those dimers to be excreted, all right? We don't want them to make it to the kidney. It can cause kidney damage. We want them to be converted back to the amino acid and reused. This is haptoglobin takes the dimers to the liver where it is converted to bilirubin. It is actually only the protoporphyrin ring 9 portion of the hemoglobin molecule that will be converted to bilirubin. Approximately 1% of the total red cells are removed from circulation on a daily basis. And then these red cells, again, are replaced by 1% of our polychromatic erythrocytes or reticulocytes. So the bone marrow is compensating for that 1% loss. Okay, and you guys all know this. What analysis can we do on a blood smear? So we're going to do our 100 cell differential of our white cells. All right, we're going to classify what type of white cells, and we're going to do our red cell morphology. Keep in mind that our red cells are not counted during the smear evaluation. We are just evaluating for size, hemoglobin content, and shapes. And then we can do our platelet estimate as well. So keep in mind, just from a smear evaluation, you can tell a lot by what type of anemia, if any, is present, what type of spleen status. If they've had a splenectomy, we might see Hal-Jolly bodies or any other type of inclusions as well. So from the peripheral smear, we can tell a lot about that patient. All right, so in lecture 10 and 11, we talked about our hemoglobin molecule. We have our heme, which is composed of your protoporphyrin ring 9 and iron. And then in lecture 11, we built our globin chain. So remember, your globin chain is going to be composed of 4 chains, 2 pairs, a alpha and a non-alpha will form 1 chain, okay? And then there's 2 chains per hemoglobin molecule, okay? And keep in mind that control of those chains is by transcription, DNA, protein synthesis, okay? We also talked about the types of chains present. Right, so remember, if you have 2 alpha chains and 2 beta chains, that's our normal adult hemoglobin A. If you have 2 alpha chains and 2 gamma chains, that's our hemoglobin F. If you have 2 alpha chains and 2 delta chains, that is our normal adult hemoglobin A2. Okay. And then also we talked about the globin chain being comprised of amino acids. One single amino acid rearrangement can result in abnormal hemoglobin, such as hemoglobin S, hemoglobin C, hemoglobin E, okay? And then we also talked about the hemoglobin oxygen dissociation curve. We talked about some reasons for a right shift versus a left shift. Make sure you guys are familiar with what can cause left shift or right shift, okay? And then what happens as those red cells are broken down? What happens to our hemoglobin contents? Remember, the body's going to try to reuse as much as it can, right? Iron is going to be reused. The globin chains will be transferred back to the amino acid, and that protoporphyrin ring 9 will be broken down into bilirubin and excreted as urobilinogen. So what's the breakdown product of the protoporphyrin ring 9? Bilirubin, which will ultimately become urobilinogen. Are there any questions? All right, so this is laying the foundation for us to move into anemias. That's what we're going to start in the next module. Yeah.
Still trying to get a handle on the action of 2,3-BPG. This exchange 2,3-BPG for oxygen on a hemoglobin molecule, is this the way that all your cells get oxygen, or is this an external control coming from a third party to that exchange? Does that make sense?
I wouldn't necessarily, it controls the shape of the globin molecule, so that better allows for incorporation of oxygen. So it kind of acts like a bridge. But once oxygen is incorporated, it is not big enough to expand the width of those globin chains with the oxygen molecule inside it. So actually that chain is broken when oxygen is incorporated. So then once the cell loses oxygen, then it can, that bridge is formed again. Does that make sense? So it can be reoxygenated. So it doesn't really,
Just wondering, if I've got a hemoglobin molecule,
mm-hmm,
it's got 4 oxygen on it,
mm-hmm,
ready to go to capture,
Mhm.
right? Is it the case of, well, I'm a hemoglobin molecule inside of an erythrocyte, I'm not giving up any of my oxygen unless somebody hands me some DPG?
no, that's not how it works. The 2,3-DPG is already there before the oxygen is incorporated. It's just like a standard structure of that hemoglobin molecule. And then once oxygen is incorporated, then the structure kind of folds on itself. It's going to do like a 15-degree fold. And then that salt ion bridge is actually going to break. And then so oxygen will be readily released when needed, depending on how it travels to the tissues, at what point is that oxygen released. Does that make sense? Kind of, sort of. Okay. That is all I have. We have our exam 2 review on Wednesday.
Lecture 11, right?
Yeah, so lecture 12 is the review. So this is the last new material for exam 2.